everyone. So in this video we'll be doing about the salient feature of DNA. Okay, so these salient features are nothing but it was also given by Watson and Crick only. It is based on Watson and Crick model of DNA only. Okay, so how DNA is structured, okay, that comes under this salient feature of DNA. So first of all, I have already told you DNA is a double-stranded structure. Okay, DNA is a double-stranded, that means it comprises of two strands. Okay, it comprises of two strands, and each strand, when we draw like this, okay, for your understanding, each strand, they are made up of polynucleotides. That's why, okay, it is a chain of polynucleotide, two chains of polynucleotides. Okay, and in that double-stranded structure, okay, phosphate and deoxyribose sugar, they forms the backbone, and the nitrogenous bases, they lies inwards. Okay, which I have already told you in the previous video also. So now the next thing, okay, now the nitrogenous bases, okay, in the two strands, they are linked together. Nitrogenous bases, that is, which are complementary, okay, nitrogenous bases that are complementary, they pair up, okay, that is, adenine pairs with thymine and guanine or cytosine pairs with cytosine or guanine. Okay, so when adenine pairs with thymine, they pair up by 2 hydrogen bond, and when guanine pairs with cytosine, they pair up by 3 hydrogen bond. Okay, and adenine are always complementary to thymine, and guanine are complementary to cytosine. Now this adenine, thymine, guanine and cytosine are nitrogenous bases. Okay, they are base pairs. Adenine and thymine make one base pair. Guanine and cytosine make one base pairs. They are called base pairs. Now, very important, since DNA is double stranded, that means it has got two strand, okay, two chain of polynucleotide chain, okay, two strands. It is not straight like that. Please remember that it is helically coiled. I am drawing like this for your understanding only. So when they are placed, okay, when these two strands are placed, they are placed in anti-parallel way. Now what do you understand by anti-parallel way? These two are parallel, okay, these two strands, okay, they are parallel, two strands are always parallel, okay, but anti-parallel means, suppose, okay, I'll show you the example. If these two are two polynucleotide chains, okay, segment of DNA, two polynucleotide chain, if one strand they lies like this, okay, and you know that it's a polynucleotide chain, so at one end one nucleotide, on the other end they have another nucleotide. One nucleotide have ribose sugar where phosphate group are free, okay, they are free, because 5-OH group of ribose sugar, it is attached to the phosphate, so phosphate is free. At the other end, okay, at the other end 3 dash OH of ribose sugar, it is free. Okay, now the other strand, they do not lies like this. If this is 5 dash OH group and this is 3 dash OH group, the other strand will not lies like this. The other strand will be anti-parallel. That means it will be like this. Okay, the polynucleotide chain in the other strand, they are 5 dash OH group. Okay, opposite to this, here it is 3 dash OH of ribose sugar, and here opposite to 3 dash OH they have 5 dash OH group of this ribose sugar where phosphate group is attached. Understood? So okay, so this is all about, you know, anti-parallel direction of the two strand of DNA. Okay, so okay, let me show you the book first for your better understanding. Okay, now all right, let me show you the book. So you can see here in the book, okay, only one strand is given. Okay, DNA is not only single strand, they have two strand like this. Only they have two strand and they pairs up like this, two strand. Now when you look into this picture, okay, here you can see 5 dash OH, this is the ribose sugar, isn't it? So 5 dash OH group of ribose sugar, it is attached to phosphate group, okay, at one end of this strand, and of the same strand at the other end we have free 3 dash OH group of the ribose sugar. Can you see that? So now this is one strand and the other strand is here. The other strand lies anti-parallelly, that means opposite to 5 dash. Here they have 3 dash OH group of the ribose sugar. Okay, then of the same strand at the other end they have 5 dash OH group of ribose sugar where phosphate is attached. So that's why it is called anti-parallel strand. DNA is double helix, okay, and it runs anti-parallelly. 5 dash OH, 5 dash to 3 dash, here 3 dash to 5 dash. Okay, all right. Okay, now another very important thing, coiling. How it is coiled, okay, how it form helix is they coiled in the helix. Okay, it is formed by right handed fashion. Right handed, that means it is coiled like this, clockwise direction. Okay, clockwise direction, not anti-clockwise direction. Right handed fashion. Helix is formed through this right handed fashion, that is clockwise direction. Okay, okay, now another thing. Now when this okay, DNA, the double strand of DNA forms the helix, one turn, okay, that means this one from here to here, one full turn from here to here, okay, one turn. The pitch of the helix, it is 3.4 nanometer. This is called pitch of the helix. Okay, pitch of the helix. So the length of this pitch of the helix is 3.4 nanometer. Okay, 1 2 5 6 7 8 9 10. Okay, all right. So this is called pitch of the helix, and it is 3.4 nanometer. And now one pitch, okay, that means one turn, okay, one turn of the helix. This is from here to here, it forms one turn. One turn of helix, it consists of how many base pairs? Base pairs, that means adenine, thymine, guanine, cytosine, like that way, okay. They form how many base pairs? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Always remember, one turn is made up of 10 base pairs, okay. They have 10 base pair. Yeah, and from here to here, okay, the length from one base pair to the other base pair, the length from one base pair to other base pair, it is this length, okay. This length, it is 0.34 nanometer, okay. So this is all about the salient feature of DNA. Very simple, isn't it? Now, okay, next, very important, it is given in your book, okay. Central dogma in molecular biology. Central dogma in molecular biology. Okay, Francis Crick, okay, Francis Crick proposed this term, central dogma in molecular biology, which tells that the flow of genetic information. We know that gene or the DNA contains genetic information, isn't it? So Francis Crick was the scientist who gave this central dogma in molecular biology, which tells that the flow of genetic information is from DNA to RNA and from RNA to proteins, okay. DNA, when it gets transcribed to RNA, and when RNA is translated into protein, the genetic information that is there in DNA, okay, it flows to RNA first, and from RNA it flows to protein, okay. And the formation of RNA or the synthesis of RNA, messenger RNA, okay, there are so many types of RNA. So genetic information flows from DNA to mRNA, that is messenger RNA. Messenger RNA. There are so many types of RNA. And this process where mRNA is synthesized from DNA, this is called transcription. Transcription. Okay. And when the proteins are synthesized from mRNA, the process is called translation. So that's why we can say mRNA is being transcribed from DNA and proteins are translated from mRNA. So central dogma in molecular biology talks about the flow of genetic information from DNA to mRNA and from mRNA to proteins. Okay, very simple. Okay, and see one thing, remember please, when DNA gets transcribed into mRNA, at first DNA, they replicate. Always remember, they first replicate. Replication means where DNA gets converted into 2. Okay, haploid numbers get converted into diploid. Okay, generating its own replica. Okay, generating the replica of DNA. First of all, DNA replicates, then only it gets transcribed into mRNA, and from mRNA it gets translated into proteins. Okay, and this happens in each and every cell, each and every cell. Okay, okay. Now, in some viruses, okay, in some virus, retrovirus, name of the virus is retrovirus. In this type of viruses, some of the viruses, okay, the flow of genetic information can be reversed also. That means from RNA, genetic information can flows from RNA, DNA can be synthesized, and the genetic information from RNA, it gets into DNA. So this type of this process, okay, it is also called reverse transcription, reverse transcription. Okay, but this happens only in some of the viruses, okay, and like retroviruses. Okay, so this is all about central dogma in molecular biology. Please do read your book. Okay, it is given in details in your book. Yeah, so this is all about central dogma in molecular biology. Okay, now coming to the length. Okay, next we have is length of DNA. So in the previous video I've already told you the length of DNA, any DNA, okay, length of DNA is determined by the number of nucleotides present in it, the number of nucleotides present in it. Okay, the more the number of nucleotides in a DNA, the longer will be the DNA, because DNA are nothing but polymers of nucleotide. Okay, it's a chain of nucleotides. Okay, so some of the example is given in your book also. There's one, okay, 174 bacteriophage. Okay, so this is the example. Okay, XII-174 bacteriophage. It is the name of a virus, the name of a specific particular virus. Okay, don't get confused. Bacteriophage are nothing. They are no bacteria. They are not bacteria. They are viruses, but the viruses that infect bacterial cell. Okay, bacteriophage are the virus that infect bacteria or bacterial cell. XII-174 bacteriophage, they have the length of DNA 5386 base pairs. Now that long, okay, that long, 1 2 3 4, when we count the base pairs in two strands of DNA, the length of their DNA can have 5386 base pairs. That much long the DNA is. Okay, it can the length of the DNA is determined by number of nucleotides, and we can also say on the basis of the number of base pairs, because how many nucleotides are there in a length, throughout the length of DNA, the base pairs will be exactly the same. Okay, number of base pair will be exactly the same, because one nucleotide always contain one base pair, base, nitrogenous base. Okay, and another is there, lambda bacteriophage, another type of bacteriophage or another type of virus. They have 48502 base pair. Just imagine that long the DNA is. Another example is E. coli. It is the bacteria. Okay, Escherichia coli, it is the name of bacteria. They have 4.6 into 10 to the power 6 base pair. Just imagine how long it would be. Okay, and talking about human being, they have 3.3 into 10 to the power 9 base pair. Just imagine human being, the length of the DNA 3.3 into 10 to the power 9 base pair. Okay, and it is the haploid number. Haploid number. So similarly, the diploid will be double of this. Okay. So now see, we are talking about DNA that is present inside the nucleus, that are packaged inside the nucleus, very tiny particles which is present, tiny organelles which is present inside the cell cytoplasm, and cell is also considered as very minute particles because we cannot see the cell with our naked eyes. So just imagine inside that microscopic cell lies the nucleus. Inside the nucleus lies this DNA. And just imagine the length of DNA, how it is packaged. Quite interesting, isn't it? How it is packaged, and it is so long. Each and every cell have this length of DNA. Each and every cell. There are millions of cells in our body, and each and every cell have nucleus. Inside the nucleus they have this length of DNA. So there must be a mechanism to for this length of DNA to get packaged, isn't it? So yes, so there is a special mechanism to pack this DNA. Okay, so in the next video I'll be doing about this packaging of DNA. Okay, thank you very much.